Experience of using chemical data, biological data, how to use that data for making decisions. And it's usually decisions how to select a compound or to understand the mode of action of a compound. And I kind of have two titles here. So the original one is the one I usually use, integrating chemical and biological data for compound selection on the one hand and mode of action analysis on the other hand. But the other aspect is I was actually compiling information about the 5 or 6 projects we were working on in recent years where we used chemical information and biological information, and I asked myself the question, so where did we actually see a signal? Where did the project work, basically? Yeah? So I'm giving an overview of a couple of projects to give you an impression of which types of information we can use in order to make which types of decisions. And so this is the outline of the talk. So first I will talk about data, so what's out there, how can we use it, but the main part will actually a couple of impressions, let's say. I will guide you through in a total of 6 projects briefly, where we use data to model cell sensitivity, for example, where we work with Johnson & Johnson, where we use gene expression data in lead optimization. We have a current PhD student together with the Sanger Institute where we correlate transcript and proteome data, and actually you see less correlation than you might expect. These projects actually led me to partially question which types of information we can actually trust, especially from the biological side, yeah, because there's really lack of understanding in many areas. But there are also a couple of case studies where I would say generally overall actually we managed to analyze data and make decisions based on those data. And that was in areas such as analyzing sleep data, read sleep data, and understand modes of action against multiple GPCRs in parallel. We worked on personalized medicine, HIV, and we also currently use gene expression data to select small molecules to differentiate stem cells, and that was working quite well as well. So I'll give you a couple of impressions of those topics we worked on. So what is the data we usually look at? It's usually 3 types of data. And so I'm a chemist by training, I'm very happy to be a chemist, but many years ago I discovered actually I also need to understand biology. If I only focus on my chemical aspects, yeah, and I don't understand readouts, what do the readouts mean? Whatever the level is, whether it's imaging or gene expression or phenotype, yeah, I don't understand that either. So it's really important for us to look at chemistry and biology. It's on the same level. I mean, it's very different aspects in both parts, but you need to understand both areas. So what we are looking at is molecular structures. We can buy 20 million compounds or so right now. We can synthesize many more. It gives us quite a lot of power. We can also store chemical structures quite well. We understand structure to a good extent. Sometimes we don't know stereochemistry, but still chemical structure we can more or less handle. But it looks very different if we go into other areas. So if we think about protein or the mode of action area, if we think about sequences, you generally have sequences of most organisms. If you look into 3D structures, our information becomes much more scarce. And the actual problem occurs if we think about dynamic interactions. So if you have a static protein, that's one thing, but the question is, in a particular tissue, in a particular state of a cell, does that interaction exist? Yeah, what are the rate constants? What are the quantitative aspects of that interaction that lead to downstream signaling? And that's really where the problem occurs. So we hardly ever have that information. So molecular structure we can handle quite well. Here some problems come up, but of course information is increasing as well. And if we think about information that links molecular structures to phenotypes, quite often we know the compound we put in, we know, for example, the adverse reaction we observe, but quite often we don't know the genotype of the patients. So if you think about adverse drug reactions, there are huge differences between Caucasian people, Asian people, and so on, and you can even subclassify those groups as well. But quite often you don't have all the variables associated with the readout. The same is true if you, for example, do high content imaging, or if you have data sets that were generated in different ways. Quite often those parameters that are associated with experiments don't allow you to pull information where you link a particular molecule to a phenotypic readout of whatever type. But that might all sound a bit too pessimistic. Of course, what is the case is our information in that triangle is certainly increasing. Yeah? And it's certainly a good time to ask the question, so how can we integrate that information, and which types of question can we ask right now? And usually there are 3 types of questions we can ask here. And usually we have information of 2 of those types, and we can ask for the 3rd depending on the data we have. And so those 3 types of questions are as follows. The first one is, if I have a particular type of chemistry, and an observation, so I know the chemistry and the phenotype in the previous triangle, and then I can ask for the mode of action based on the data I have. For example, chemical data, I can put in, I will give an example of that later, I can put in the molecular structure, I can anticipate targets, predict targets, and I have a mode of action hypothesis. That's the first application of the types of data I've shown. The next one is the question, which compound should I actually make to achieve an effect? So here I'm starting with the phenotype, I go back to pathways that are involved in a disease, let's say, or stem cell differentiation, whatever I'm interested in. I go back to the targets, then I ask the question, what do I know about chemistry targeting those targets? And then I can design, for example, a pathway-focused library. So I go back to the chemistry based on the information I have again. The third type of question is, does a certain patient respond better to a drug than another patient? Or in other words, what's the right drug for a particular patient? So in this case, I want to have a phenotype change. I have different options of chemistry to put in. And then the question is, what's the genotype of the virus, let's say? And then I can say which chemistry would ideally be matched to a particular patient, depending on the genotype information that I have. And so we can ask for all of those three variables in the triangle. And I will give examples of all three of them in a couple of slides later. So in the group, so it's about 22 people, it's quite diverse. So we analyze chemical data, we have people from biology, pharmacology, people that do computer science. And what we also do is we work quite a lot with pharma companies or other companies as well in the life science area. The reason is in academia it's always nice to publish a paper, but I found it also quite motivating to see what we do really used in practice, in a practical setting. So the proof of the pudding, as you might say, is in the eating, so, and in the application in our area. So that's certainly what we do as well. What we do generally in the group, it's quite a variety, I might say, of research areas. So mode of action analysis, what we started with, and it's still a third or so of the work we do in the group. It can be applied in quite a, I was surprised about that as well, quite a wide variety of areas. You have dozens of settings, dozens of areas where you have links between chemistry and, let's say, an adverse drug reaction or a high content screening readout. Or you have a compound mixture where you see an effect in a particular cell system. You can do analytical chemistry to find out what the ingredients are, and then you can understand the mode of action and so on. That's really applicable in a surprising number of cases. What we started doing 2 years ago is model compound mixtures. So that's interesting from the safety perspective as well as the efficacy perspective. So when it comes to compound safety, quite often we don't understand single compound effects. But of course, if you have either consumer products or you have therapeutic agents, quite often what you're interested in is combination effects. You have if people get older, they take 10 drugs, or they take a huge number of different drugs at the same time, and you don't really know how they interact. So we're trying to model that, but we're only one step along that line. It's really quite a long way to go, I should really say. And finally, what we moved into now, more recently, is looking into transcriptomics readouts, because I think that's actually quite a good, price to signal trade-off is actually in quite a good position right now. So you have gene expression data for large numbers of compounds as well as diseases. So it's a good time, I think, to analyze that type of information. Of course, the question is, which signal can you see in there? That's always what you have to keep in mind. Does transcriptomics actually tell me something about my question, how to answer that? Quite often the answer is no, but sometimes the answer is yes at least. So that's what we do in general. And what I will go through is now a couple of case studies where we use chemical information, biological information to model bioactivities in order to, build a model that allows us either to pick a better compound or understand what compounds do. So the first three cases that led me to question a little bit, do we actually have a signal in the biological data that we want to model? Are those three cases here. We were modeling cell sensitivity, so compounds were used to treat cancer cell lines, cytotoxicity was measured, so GI50 values, and we tried to model those values on a large scale. We used gene expression data for lead optimization. So here the question was, if I have different lead series, different scaffolds, can I use gene expression data to say this series is better than another one, and which compound to pick from that series? And this is a project we currently have with Sanger, where we look at different levels of biological information. Okay, so if you think about the first project. So what we did here is we wanted to model cell sensitivity, and so we took data from the NCI60 cancer cell line screens. It's quite large-scale data that was provided by the NCI for more than a decade by now. It's a large data set, so we have many compounds that are tested on 60, as was originally thought, but it's 59 as genetic analysis proved later. Cell lines, and now the question is, can we model that type of information? And in practice, of course, not about cancer cell lines, because what you would like to do is actually you have a primary tumor, so you're going to patients, but the question is, can we develop a general concept that we can model cell sensitivity based on biological readouts? So the idea is to translate that to patients later, of course. And what we did here is, so we took data from the NCI60 cell line screens, and 2 years ago or so, the Broad and Sanger characterized the NCI60 screening deck on a variety of levels. So sequence data was around before already, but then they took transcriptomics data as well as proteomic data. You can measure whether transporters are expressed and so on. And the question is then, in some cases you know what drives the cancer, but in some cases you don't quite know. And so we attempted to model cell sensitivity across cell lines for a large number of compounds. And so that is displayed here. So it's about 20,000 compounds we had on the set, as well as 60 cell lines. And this is kind of an integrated chemical and biological space that we attempted to model. And you might be familiar, and that's the work of Jarl Wigbig here as well, of the Protea chemometrics modeling approach. It's actually very similar to that. Yeah, you have variants in chemical space, variants in biological space, and you try to model that in an integrated model. The original PCM approaches were often focused on binding site information, yeah, but that's replaced here by some type of biological descriptors, but still you model variants in both spaces and you try to correlate that with a signal, yeah, with a dependent variable, with a readout. And so how did we model that information? So we had a block of chemical information, biological information, and the readout. And so what we use from the chemical side, and that's quite often what we use, is circular fingerprints. It's a very simple descriptor. It's nearly too simple to be true, but it works remarkably well. And the way it's constructed is you have a heavy atom, you have a molecule, and you look at all the heavy atoms in turn. You type them, for example, according to hybridization state. In this case, it's actually MOL2 force field atom types. So you look at every heavy atom in turn, its neighbors and its neighbors. So you can construct a count vector for every heavy atom in turn. So you do it for this carbon, then for that one, and so in this case you get 9 or 10 or so different patches that describe that molecule, and they work remarkably well. And so I worked with those descriptors for a number of years now, nearly too long. And so the reason why I think they work quite well is actually they are abstract enough, since you lose connectivity between the layers. So you don't keep the information which atom in the second layer is bound to which atom in the first. You only know those atoms are on the first layer, these ones are on the second layer. Yeah? So it's to some extent it's abstract, so you can generalize. But you keep the atom types quite concrete. So you don't use, let's say, electronegativity or so. Yeah? You actually have quite concrete hybridization state-based atom typing. So it's the right level of being concrete enough, but also abstract enough. Yeah? There's an optimum somewhere in between. And the other aspect is actually disjoint but overlapping features. So disjoint, they are independent features, well, according to the way they are generated, but of course they overlap. So you look at this atom, its neighbors, as well as this atom and its neighbors. They are formally independent, but of course the atom-to-atom connectivity overlaps between the features. So again, you don't have a descriptor that spans the whole molecule, but the patches taken together describe the whole molecule in an overlapping way. So I think it's again the right level of being focusing on local connectivity, but still keeping long-range distance information into account as well, but not explicitly, only implicitly. Yeah? So I think in both of those aspects, those descriptors actually are they're quick to calculate as well. They only depend on 2D connectivity. So they actually represent quite a good trade-off, I think, when representing molecules in a cheminformatics way for modeling. So this is how we quite often actually describe our molecules. And from the biological side, we actually had quite a variety of information available for all 59 NCI60 cell lines. So that ranged from gene transcript levels to miRNA expression, copy number variations, also transcriptome, also transporter expression, and so on. And so we tried to integrate that. So and it was a total of 17,000 compounds, close to 2 million data points. And so we did different types of validation. For example, we tried to predict cytotoxicity for a cell line we left out. It's quite important not to fool yourself into believing that your model works. You always need to make sure that you have a validation set, you would call it, that you can trust. So where you nearly force some distance between what you put into your training set and what you evaluate your model on. In our case, we could leave out cell lines, we could leave out clusters of compounds, so we really force a validation set that's far enough away from your training set. And that's as good as you can do in practice. But of course, the real proof is then in a real prospective validation. That's what we are currently doing as well. Okay, so there are two conclusions from that model that I would like to share. The first one is, so here, that's the predictions of a leave one cell line out validation against a particular cell line. The particular cell line is not the main point here. The point I'm making is, overall, from that model, we were actually able to model cell sensitivity to a surprising extent, also if we extrapolate to a new cell line. So that's the good news, so to speak. But what we also did is we compared whether different types of biological information give us a different quality of our models. And what you would expect is that you have certain types of biological input that actually give you a much better signal for modeling cell sensitivity. But what we've seen here is whether we use gene expression information or transporter information or copy number variation or whatever type of information we use from the biological side, the RMSE of our model is actually pretty similar. It's always between 0.56 or so to 0.67. So it's actually a pretty narrow range of models we're getting. And that is surprising. So I don't think that we can say that we have that we can model cell sensitivity across cell lines, across mechanisms of action. It's a very global model. Because otherwise what you would see is that certain types of biological information give you much better signal than others. It seems that all of those descriptors describe biological variants, so we can model variants, but we would need to look into the features to really see whether our features mechanistically make sense. And I think we are not there yet, at least we are not. There's still more work to be done. So sometimes you get a good model, but if you look into whether you can trust the model, it really describes what you want to model, then sometimes that can be more questionable. That's what I just summarized. Okay, so that was the first project. The second one was where we used gene expression data in order to pick compounds and lead optimization. So that was a large project with Johnson & Johnson that was just published as well on Drug Discovery Today. So what we had was a prospective model, a prospective project, where we looked at 8 projects at Janssen, and we asked people, so do you have any current projects where you think gene expression data could be of help in order to pick a particular series of compounds? And to say, okay, series A is better than series B, so what's the compound we should continue with, that we should optimize, and in which way should we do that? And that was a real prospective project, which was not trivial in some cases. Because we needed to really go with the project. If you do it retrospectively, it's easier to evaluate whether something worked or not. But we really had to wait for new compounds to be synthesized and so on. But that actually improved, I believe, the quality of the study. And so we looked at 8 different projects. So it was projects working on different targets, different disease areas as well. Oncology was quite prominent though. And all of those projects looked very different when it comes to the gene expression you can detect. And that is something that's a bit, you never quite know what you get if you look at gene expression data. Sometimes you get too much of a signal, or actually, which you don't even understand. Sometimes you don't get any change or hardly any change, so no response on the gene expression level. You never quite know what you're getting here. And so in this case, you also see that sometimes you have thousands of genes changing more than one log-fold. Sometimes you're at a very small number of genes. And so in all of those areas, we asked the question, can we use gene expression data to pick a better compound? And I'll give you one example of where we believe this approach was successful. So here you see for a particular project, you see a set of particular compounds in a given stage of the lead optimization. Every color is a different scaffold. The bars are the numbers of gene expression changes, and the dots is the on-target activity. And so what you would like to do is, of course, you want to have, in this case at least, you want to have high on-target activity, but at the same time you want to have compounds that are not too promiscuous, that don't change too many other parameters in your cell. So that means you would like to have those dots very high and those bars pretty low. And if you only focus on target activity, usually what you would do is you would probably pick compounds from the green series. They are more potent than, for example, the compounds from the red series. But what we have done here is we actually focused on both efficacy, so on target activity, as well as the side effects or potential side effects of those compounds. And then actually that led us to believe that maybe the red compounds are a better choice. They're not as potent, but they have fewer side effects, as we anticipated at least from the gene expression profile. And so in the next series, that was the next series, compounds were optimized, and so we were able to optimize on-target activity while keeping this low side effect profile. Okay, so we think that in this case gene expression data was useful to pick compounds based on both criteria, on-target activity, so which is then efficacy related, as well as the off-target activities, so side effect related parameters. Okay, so this is, I think, where gene expression data was useful in the end. And so overall, so it was 8 projects. So we've seen in 2 projects gene expression data was able to support no-go decisions. In 3 other projects that was a bit mixed. And in 3 other projects again there was no clear signal. There also seems to be, and that's maybe not too surprising, a relation to the disease areas. So in oncology related projects where you hit kinases more often, where you see a signal later in the signaling cascade because you hit some transcription factors, you see signals more often. So there seems to be a relation to disease areas as well. So that was the second example. And that's one project where I cannot say yet, I can't say much about right yet because it's still ongoing, so we still need to compile all the information. So that's a project with Sanger where we look at osteoarthritis patients. And in the first study it was only 12 patients, and we were looking at, or we are looking at correlations between RNA levels and protein levels. And sometimes it's really interesting if you have some, you learn textbooks when you study in the first year of your studies, and you have those paradigms, DNA goes to RNA goes to protein. That's what you learn, and maybe that's even true in the majority of cases, but quite often, very often, that's actually not true. And so sometimes you see quite a lot of RNA that doesn't get translated into proteins. You see the assumption is that you have particular transcripts that are tissue specific, variants of genes. But also that's not quite the case when we looked at the data. So it's really difficult to say whether this is true in a particular case or whether it's not. And that gives us a different problem, of course, because the question is, if we want to model data, analyze biological data, what actually gives us a signal? Is the signal really in the RNA, the transcriptomics readout? Maybe sometimes it is, sometimes it's not. So this is still largely ongoing work, but so far I found the results from this project shocking, so we will present that in more detail once we have all the data available. And one reason I think is quite often the data we generate from the biological side is really technology driven. So really the question is more, what can we measure? Let's measure it. Let's say there's the 100,000 genomes project in the UK right now, and of course there will be some signal, if you have a link between disease and the sequence, but sometimes that will actually, the signal will only come in much later. I mean, you have transcription, then translation, you have post-translational modifications, you have dynamic interactions and so on between proteins, you have location and time elements, all that is of course missing. And then the question is, where do we really have the signal? In our case study from the using transcriptomics profiles for lead optimization, well, in half of the cases we've seen a signal, in the other half we didn't. So in this case it was about 50-50. Okay, so these cases maybe didn't work as well, but we also have a couple of cases where I believe a data analysis of chemical and biological data was able to help us either pick compounds or understand what compounds do. So the first project was understanding red sleep data. So that's a project with Eli Lilly, and new sleeping pills are still of current interest. Not because it's difficult to put people to sleep, that's actually quite easy, but the problem is more side effects. So you wake up and you feel drowsy. And that's still an incentive for some companies, such as Lilly, to work on sleeping pills right now. So they had a data set of 500 compounds where rats were treated with those 500 compounds. You can measure how well rats sleep. You can measure electroencephalograms. You can measure how much they move around if they are awake and so on. So you have a profile for effect, good sleep, and for side effects, which means if the rat is awake but it does not move around, it does not eat, then it's drowsy. So you can measure efficacy as well as side effects. And the question was, how can we actually understand those bioactivity profiles? And what we did here is we used target prediction models, which we used for quite a number of years in our group, and which also many other groups, of course, have worked on. And so we use data such as from Campbell, so the large database at the EBI, that contains millions of compounds that are active on the order of 1,000 or so human protein targets. And that's information we can compile into target class models. So we use all of those links between ligands and targets. We build models for every class. We can put in a new compound and we can predict new targets. So how does it look like in practice? From the compound side, we again use those circular fingerprints. That's what I've gone through before already. And how do we classify our compounds? So I learned in my PhD that sometimes it's good to really stick to a simple model. Because there was one approach where I spent half a year on doing something very complex and really gave mediocre results. And since then I realized, okay, sometimes it's good to really keep it simple. And so we use something that's called the Base Classifier. Something you might know from spam detection as well. It's a very simple concept. You have two classes, so you have active and inactive compounds, or emails you want to read and spam emails, and those emails or compounds contain features that are associated with your activity or inactivity. Same with the words in the emails you receive. So if emails you want to receive, they show those features. So these are the features in active compounds. And you have emails you don't want to receive, spam emails. So these are the features in spam emails or inactive compounds. And you just model probabilities in those classes. Again, it's a very quick way to learn. You can model millions of data points in hours, or it depends on how many dimensions you have in your feature space. Okay, so what can we do then? So we can put in compounds and we can predict targets. And you see from those predictions, you see what are likely targets, and you also see how promiscuous your compound is likely to be. So here, for example, Gleevec is a much more selective compound than reboxystaurine that really hits all the isoforms of protein kinase C and so on. What you need to keep in mind is those models are based on data. If you don't have enough data, your model will predict a very selective compound, but it's actually due to a lack of data on this chemical space or on other targets. So it's very important to assign confidence intervals as well. That's not shown here, but that's what we do as well. And those predictions we can apply to those 500 compounds that were modeled in rats, well, they were tested in rats, and we wanted to understand what are the bioactivity profiles that are associated with good sleep and either no sleep or side effects. And so we put those bioactivity profiles in decision trees, and you read those from the top. So you have a combination of targets that gives you an effect. So red here means these are compounds that show the desired effect. Blue means these are compounds that either don't show the effect or that show side effects. And so that means for the whole data set you get a list of different bioactivity profiles that are associated with your readout. So what that means is we can analyze or predict modes of action of GPCR active compounds, and we can simultaneously consider multiple mode of action hypotheses as well as multiple targets in every mode of action hypothesis. And that's really what you would need if you have CNS active drugs, antidepressants or anti-anxiolytics and so on. You actually need this type of bioactivity profile against multiple targets in parallel. And that's what we prospectively validated. So we looked at 21 and we validated that on the target level as well as on the phenotypic level, and so on both levels that was actually pretty successful. And so I will think I have only very brief time to go through the other two examples. So one project we worked on is on modeling HIV compounds that are active against HIV. And so in this case, we again used the Protea Chemometrics related approach. So we modeled a set of compounds against particular mutants of reverse transcriptase in HIV. So we had a large number of compounds against different mutants of the enzyme, which is equivalent to new patients that come in that can get sequenced, can get the virus sequenced, so you can determine which drug to apply. And so we modeled that on quite a large scale. So we used tens of thousands of sequences that were provided by Tibotec, as well as then hundreds of thousands of compound mutant pairs. So we were trying to predict if a new HIV patient comes in and gets sequenced, can we predict which drug that patient should use? And what we see is, so if we have many data points, we can actually model efficacy in this case against a particular virus mutant. If we don't have enough data, we cannot model that. And that's really common, the better. That really, the number of data points makes a huge difference in the models we generate. And the final model is, the final approach is where we use gene expression data for compound selection. So that was done by a very able PhD student in my lab, Yazaman. So the idea here is quite simple. If we have compounds that are linked to a change in gene expression, and we have diseases that are linked to a change in gene expression, then we can hypothesize if the compounds induce the opposite change in gene expression to the disease, then there might be a link between disease and that compound. It's not yet a mechanistic hypothesis. I wouldn't go that far, but at least there's a signal that maps both spaces. And so we use different databases that contain tens of thousands of compounds, and we prospectively validated that in different areas. And that's cancer cell lines as well as stem cell differentiation. And so what we found very encouraging is that we find a large number of compounds that are active, and they are not only active, they induce apoptosis in cancer cells, but they also do so in the desired way. So in the gene, if you select compounds based on gene expression, you have a mode of action hypothesis. So you can say which pathways you want to hit, basically. And then if you do cell cycling or different biological analytical methods, then you can see whether your compounds induce a biological effect along the desired way. So I don't have time to go into detail here, but that's one of the validations we did. And the other one is to differentiate stem cells. And that's what I found quite fascinating, actually. If you take stem cells, you select a small molecule, then after 2 weeks or so, the stem cells differentiate and you get cardiomyocytes, so really beating heart muscles, basically. And I found that fascinating to see. That was really reminded me of the old times when I started studying. The chemical matter you select can really change biology. Otherwise, that heart muscle basically wouldn't exist. So we started a company based on that as well. So we try to repurpose drugs that are used for common diseases for rare diseases, and that's what we try to extend right now. It's going quite well. It's a good environment in Cambridge to do that. Okay, so where are we with data and drug discovery today? There's quite a lot of data we can use. We can use it successfully to select compounds and understand modes of action. And I think where there are clear links between a feature and effect, for example, predicting the efficacy of HIV drugs, we can actually generate pretty good models. It's always important to, well, have a good reason why you include particular features in the model. But we should also be realistic. What's in the data? Not everything will be in the genome and so on. You also have to see where your signal might be. So thank you to all the people we work together with, and thank you for listening.
Thank you very much, Andreas. We were actually 5 minutes after lagging behind the schedule, and I think that there is time for questions. Are any questions? Yes.
So when you build your base...
Can you please use the microphone? There is a microphone here, it's very difficult to hear.
About when you build your Base models, whether you have a minimum number of compounds that you use for each of the different targets, and whether if you have some targets where you've got lots of data and other targets where you've got a small amount of data, does that make any difference to whether you're likely to get a target with a small amount of data predicted as, an active against a new compound?
The first part of the question, we have a minimum number of compounds. It's usually around 20. Below that, we don't build any models. The other criteria are based on organism. We usually have a 10 micromolar cutoff. These are the other criteria. And the number of compounds makes a huge difference. And that's one of the tricky parts, because it's really the trade-off of using more information versus having a bias between the different submodels you have. Yes, more compounds make it much more likely. to hit a target. It's in particular, you have the problem if you have related receptor subtypes. Let's say serotonin receptors, you have many subtypes, and you have ligands for one receptor where you have 20 ligands that are very similar to another receptor where you have 1,000 or so ligands. That really dominates your model. You hardly ever predict the small model. Yes, we have that problem.
I wondered if that was the case.
Yeah, we do. We tried removing compounds, sampling compounds and so on to arrive at some kind of best solution, but it's not optimal. Yeah, we have that problem.
On your decision tree, where you're looking at all those targets with the sleep, are you actively looking at PGP efflux? Because if the compound goes into the brain and then is flushed out, it will have no effect.
Yeah, that's something we tried to do. We did not on the current models. We tried to include that. It did not change the accuracy of the predictions. Yeah.
It's quite important.
Yes, it is. Yeah.
Okay. Are there any more questions? I have also a question, maybe a little naive, but there are many people here who are not familiar with drug design and the process of doing it. So you mentioned in one of the slides, that I didn't fully follow, that you optimize against target and against side effect. And actually, what is the process of this optimization? How do you see it?
Well, so in this case, every project was a bit different. So in this particular case, we had two different assays. One was target related, so that's where we wanted to increase activity. And the other one was what we assume to predict side effects is the kind of how many changes do you have in the gene expression profile. But of course, it's an assumption that that is actually predictive of side effects. Maybe it's not. But we had those two different types of assays where we tried to optimize into different directions. So increase activity here and decrease activity here. In this particular project, but it was different in every project. But that was the process here. So multiple selections where we picked opposite trends in those two assays, basically.
But are they actually based on some models of Qs or?
No, that was experimental data in this particular case. Yeah.
Are there any more questions? Otherwise, yes?
The current research project, like DNA to RNA, RNA to protein, do you consider in which microRNAs is also necessary in that process? Yes. And how about the epigenomics? Because right now epigenetics is also concerned with the DNA to RNA protein. And microRNA therapy is currently going on processes. So, did you focusing on microRNAs in this project?
MicroRNAs as well, yeah, that is true. But for details, I probably need to leave it to a later stage to give the full overview of the project. We started quite recently with that. MicroRNAs we look at as well. Yeah.
Thank you.
Okay, thank you, Professor Bender, for your talk.
Okay, thank you.